Okay, I'll share my screen.
Okay, ma'am.
There will be board available here, but I can't see it now. So I'll be sharing my screen. Yeah. You can see the whiteboard, right?
Yes, ma'am.
Mm. So first today we will be discussing about the structure of autumn. Structure of autumn, chemical bonds, and the molecules. What do you mean by atoms? What do you mean by atom? All the living organisms are made up of matter, right? All the living organisms are made up of matter. So, all the matter is made up of your atoms. Matter is made up of atom. Got it? So, what is the structure of atom? What is structure of atom? First, a atom is made up of a central nucleus and outer shells. Got it? So the central nucleus will contain your neutrons, protons. What is meant by neutrons? What is meant by protons? Any idea? Neutrons have the neutral charge. It will be having neutral charge where your protons will be having positive charge. Today you will be knowing some terms related to your atom like atomic mass, what is neutral atom, what is atomic number, how it is denoted, and mass number, how it is denoted. So first coming to your atomic number, you say that carbon, hydrogen, helium, lithium, beryllium, boron, right? They got different atomic numbers. Do you know how these atomic numbers are given? Any idea how these atomic numbers are given? I said that protons, right? It
Based on the
contains
count of
protons. So atomic number is given based on number of protons. Protons. So in the hydrogen, how many protons are there? You already gave the answer. So I said that based on number of protons, atomic number will be given. So in the hydrogen, according to us, according to your formula or the line which I have stated, we can say that there is only one proton in the hydrogen. Coming to helium, there are two protons. So based on the protons, your atomic number is decided. Okay. So what is mean by neutral atom? What is mean by neutral atom? Any idea? I said that protons will be having positive charge, right? where neutrons will have neutral charge and electrons will be having negative charge. So neutral means without having any charge. When there is equal number of, or when the number of your protons and number of your electrons are same, it is known as neutral atoms. Okay, got it? Example, oxygen. Oxygen will be having same number of protons and same number of electrons. So it is electrically neutral. Got it? So next is your atomic mass. Coming to your atomic mass. What do you mean by atomic mass? Atomic mass is denoted by number of protons plus number of neutrons. Okay? And you should be able to differentiate what is mean by atomic mass and mass number. Is there any difference between this? Atomic mass and mass number. Mass number is also like total number of protons plus total number of neutrons. But the only difference is the mass number can always not be an whole number. But coming to your atomic mass, you can say that it will be always in the whole number. Got it? But coming to your mass number, based on your naturally occurring isotope of the elements, they will be in the form of decimal values. Okay. And these both are designated by my units are AMU. Got it? So today first we discussed about the structure of atom. Coming to structure of atom, it is made up of nucleus and external shells. So nucleus will contain neutrons and protons, where the shells will contain electrons which are revolving around it. So neutral is in neutral characteristic means neutral charge, where proton will be having positive charge and electron will be having negative charge. Okay, protons have positive charge, electrons have neutral charge. So what is mean by atomic number? Atomic number is designated by Z, and based on number of protons, your atomic number will be designated. And what is meant by neutral atom? When there is equal number of protons and equal number of electrons, it is known as neutral atom. Coming to atomic mass. What is mean by atomic mass? When number of protons plus number of neutrons will give your atomic mass. It will be measured in AMU. Got it? Coming to mass number, they will be in the form of decimals. Okay, completed. Next, we are going to discuss about what are isotopes. I said that if you are okay, you can write this line for atomic mass. Average mass of all naturally occurring isotope of an element. I said that different types of isotopes, right? So when you see the carbon will be in isotope of 12 and sometimes isotope of 13. So when you see the proton and neutron, we see here the protons will be 6 and also neutron will be 6. Sometimes here the protons are similar but the neutrons will be differentiated. Got it? In the isotopes, your protons will be same but your neutrons will be different. Keep it in mind. Proton number does not change, but neutron numbers will be changing in your isotope. Okay. Then you will be discussing about your bonds. What is mean by chemical bonds? How many chemical bonds do you know? Chemical bonds can be designated as, say, how many types of chemical bonds? What is chemical bond? Students, you have to answer.
Ionic bond, covalent
Okay.
bond,
Okay. And in naturally occurring any system, I said that atoms, right? Atoms will become molecules. So this molecule should have certain bond to hold them very close, right? So there are different types of bond, and based on bond strength, they are divided into strong bond and weak bond. Any idea about strong bond and weak bond? So your ionic bond, which you always know and which is very easy. Someone is touching my board, maybe. Ionic bond, covalent bond.
hydrogen bond,
I am asking about strong bonds,
metabolic bond.
metallic bonds. Okay. So your chemical bonds, based on your strength, they are divided into strong bond and weak bond. In strong bond, there are three bonds that are ionic bond, covalent bond, and metallic bond. Coming to your weak bonds, you know very easily. Dipole-dipole interaction. Any idea about dipole-dipole interaction? Hydrogen bond, your van der Waals forces. Maybe you have studied in your
Sigma
intermediate, right?
bond,
Sigma bond, pi bond, which are present in your hydrogen bond, right? So, first coming to your ionic bond. What do you mean by ionic bond? It is formed due to complete transfer of electrons. When there is a complete transfer of electrons, you will be forming cations and also your anions. They will be attracted by electrostatic force. What is meant by electrostatic force? Any idea? When there is a positive and negative charge, there will be an attraction between them, which is known as your electrostatic forces. Okay. Example, coming to your ionic bond, coming to your ionic bond, example is NaCl. Do you know what is the charge of NaCl? Na plus Cl minus. Due to complete transfer of your electrons, you will be producing this NaCl, right? So your valency is 1. So it requires 1 electron. So one atom is helping to give another atom get an electron. So it will be possible between your metals and non-metals. Okay. Metals and non metals. It will be having high boiling point and melting point. Okay. High BP and MP. Got it, Nana? Hello.
yes,
Can you
ma'am.
understand? Mm. So, mm. First is ionic bond, which is formed due to complete transfer of electron, due to which you will be getting your cations and anions. So, it will be possible between the metal ions and non-metals, and it will be having high melting point and high boiling point. Got it? So, next is your covalent bond. Maybe I can change my color. So next is your covalent bond. Covalent bond is formed by any idea? Covalent bond. Covalent bond is formed due to sharing of electrons between your metals or non-metals. Okay, mostly it is between the non-metal ions. Okay, they will be having poor conductivity of the electricity. Got it? So in the covalent bond, it is of 4 types or it is of 6 types. Okay, first is single bond, second is double bond, third is your triple bond, fourth is your polar covalent bond, fifth is non-polar covalent bond, sixth is coordinate covalent bond. Someone is touching the board, so please don't play here. So I said that covalent bond is formed due to sharing of electrons between two atoms, right? So it will be very strong because they are trying to share, right? So there are 6 types: single bond, double bond, triple bond, polar covalent bond, non-polar covalent bond, and coordinate covalent bond. So first coming to single bond, only one electron is shared, due to which only there is a bond. Like example, your hydrogen. Hydrogen will be having one valency, right? So it will be sharing with the another hydrogen. So both will become stable and it will form H2. Coming to your double bond, example is your oxygen. Oxygen will be having one pair of electron, one lone pair, right? If it pair with the another oxygen, it will be forming another pair. So now what will happen? Oxygen will also become stable, O2. So coming to your nitrogen, the valency is 3. So it can form 3 bonds. It will be having 3 lone electrons. So it will bond with the another 3 lone electrons of your another nitrogen and form a N2, right? So coming to polar covalent bond. What is polar covalent bond? The electrons will be shared unequal because one atom will be electronegative. Example, your HCl, okay? H plus Cl minus, right? So this polar covalent bond is formed due to, it is also sharing of electron but unequal because one of your atom is more electronegative. Got it? And coming to non-polar covalent bond, here the sharing of electrons are equal. You can write your single bond, double bond, triple bond in your non-polar covalent bond. Coming to coordinate covalent bond, here what is happening? Example, I will be taking NH3, okay? NH3. Okay. Here what will happen? The nitrogen will try to donate lone pair. Okay. It will try to donate lone pair to your hydrogen. Okay. So it is called as Lewis base. Any idea about Lewis acid and Lewis base? The atom which has ability to donate lone pair of electron is known as Lewis base. The atom which has the ability to accept lone pair of electron is known as Lewis acid. See here, our nitrogen is trying to donate one electron, so it is known as Lewis acid, where hydrogen is able to take electron, right? So now I'll show you one example. NH3 plus BF3 will give F3B. Okay. Here your NH3 is trying to give electrons to your B. So it is known as Lewis base, where this molecule is trying to accept your pair of electron, lone pair. So it is known as Lewis acid. Got it? So first we discussed about the ionic bond. Next we discussed about the covalent bond. Covalent bond is sharing of the electrons between two molecules, and it will be mostly occurring between non-metals. It will be having poor conductivity of the electricity. Then there are 6 types. First is single bond, second is double bond, third is triple bond, fourth is polar covalent bond, fifth is non-polar covalent bond, fifth is coordinate bond. Okay. Single bond, when there is sharing of only one electron. Second, sharing of 2 electrons. Triple, sharing of 3 electrons. When their valency, based on the valency, we will say single bond or double bond it's forming. Okay. Coming to polar covalent bond. Polar covalent bond is formed due to sharing of an unequal amount of electrons, because one molecule will have the electronegativity more. Coming to non-polar covalent bond, the electrons will be shared equally. Example, your single bond, double bond and triple bond. Coming to your coordinate covalent bond, it is possible between the Lewis acid and Lewis base compounds. Okay. One is able to donate your pair of electron and another is able to accept your lone pair of electrons. Got it? And keep it in mind that ionic bond and covalent bond are intermolecules, means within the molecules. Okay. Then what do you mean by electrostatic attraction? Any idea? You have to remember all terms because it is very important. Got it? Electrostatic force. It is the attraction between charged particles. Okay. Got it? What is meant by electrostatic force? I said that electrostatic force is possible between two molecules, right? Okay. So first, hydrogen bonding. Next, we will see about the hydrogen bonding. So hydrogen bonding is a, first we discussed about the strong bonds. Now we will discuss about the weak bonds. Okay. So coming to weak bonds, first is hydrogen bonding. Uh, do you have any idea where we can see these hydrogen bonds? Coming to your DNA, all your nitrogen bases will have the hydrogen bonds, right? And do you have any idea why water is in liquid condition, not in gas or not in solid state? It is all because of your hydrogen bonding. Here, if you want, you can write this definition. Hydrogen will be attached to the higher electronegative atoms or electronegative atoms having lone pair of electrons. Okay. So, how it will be generally represented? Hydrogen can be attached to your oxygen, or other than the oxygen, it can attach to more electronegative atoms or pair of electrons. Got it? So, you can represent it in the form of, if it is hydrogen, you can form with B. Here, A represent hydrogen which is attached to A or B. A will be like more electronegative atoms like nitrogen, oxygen and fluorine. Coming to your B, it is also electronegative atoms like nitrogen, oxygen or fluorine. So, this hydrogen will be very stronger than van der Waals force but weaker than covalent bond. Okay. I'll show one phenomena. First, when there is an OH, O, okay, hydrogen positive ion will be donated to your oxygen because it is more electronegative, due to which it will be getting partial positive charge and it will try to attract your electrons from your oxygen, negative charge from your oxygen, okay, due to which your hydrogen bond will be formed, okay. Then coming to your another force called van der Waals force. It is the weak attraction force between molecules caused by the permanent or temporary dipole moment. Got it? This is all about your atomic molecule structure. Then again, we will discuss about the principle of biophysical chemistry. Got it? So, what is mean by biophysical chemistry? Any idea? Here, you will be discussing about some key points like pH, you will be discussing about pH, your thermodynamics, reaction kinetics, enzyme kinetics, and colligative properties. Okay? So, first, what is meant by biophysical chemistry? It is the physical principle to understand biological system. Okay. You will be seeing your physical principles or you are taking the help of your physical principles to understand your biological system. Okay. Then what is the I am saying that study about the biophysical chemistry. What is the use of it? You can understand your cellular homeostasis and also understand enzyme function and also metabolism and your physiology. Got it? So first I said that you will be studying your pH, right? What is mean by pH? What do you know about the pH? It is a negative log.
Neutral.
Yeah, you will be having acidic condition, neutral condition, and basic condition. What is the definition? Can you say? It is an negative logarithm of hydrogen ion. Okay, you can give definition as pH is the negative logarithm of hydrogen ion concentration. Got it? And coming to your pH scale, as you said, coming to your pH scale, if your value your value will be lies between 1 to 14. Got it? So first, if your value is less than 7, it is called as acidic in nature. If your chemical is less than 7, it is known as acidic in nature. If the value is 7, it can be called as neutral. Got it? If it is less than 7, it is known as basic. Okay, students? So you can just by saying this, you can say that only 1 unit change in the pH can do 10-fold change in hydrogen concentration. Keep it in mind. 1 unit change in your pH can represent 10 folds change in hydrogen concentration. Got it? Okay. Next, what is your biological importance? Any idea? Any idea about biological?
Ma'am, once repeat this 1 unit change in 10 folds, it is not understood.
If there is 1 unit change in the pH, it will cause you 10-fold change in the hydrogen concentration. Okay, I'll give you definition. Write this. If there is 1 unit change in pH, if your unit is changing, if it is more than 7, it should be in SD condition. If there is only 1 unit change, what it will become? It will represent a 10-fold change in hydrogen concentration. SD contained Nanna. Mostly H plus ions untaya, OH minus ions untaya. Hello, students. An acid will contain or which will release more hydrogens in water, right? Where base will contain OH. Keep it in mind that acids will contain more hydrogen. Okay? Where base will contain OH. Got it? So what is the examples of acid? Example HCl, H2SO4. These all are acids. Where coming to strong base. What is base? NaOH, potassium hydroxide, all comes under base. Okay. So acids will be releasing hydrogen or acids will contain more hydrogen atoms where base will contain more OH ions. So I am saying that when there is only small change, your hydrogen ion concentration will also change. Okay. Okay, I'll take it as okay. Then coming to your biological importance. There is there any use to study about the pH? Yes, there is use to know the enzyme activity. Okay. So all your enzymes will be working at their optimum pH. We say that we don't know what it is, but we say enzymes will work at say optimum pH range. So enzymes are made up of proteins, right? Enzymes are made up of proteins. So proteins are made up of amino acids. Okay. So coming to your proteins, proteins can get denatured at high temperature or high pH, right? So the enzymes require optimum pH to survive and to do their function. What is the function of your enzymes? These enzymes will help in your metabolism, right? If there is no metabolism, what will happen? The body will become equilibrium. It leads to death of your body, right? So it helps in metabolism. What are the metabolic cycles or metabolic pathways in your body? Glycolysis, citric acid cycle. You have been studying till your since your Krebs cycle, citric acid, oxidative phosphorylation, TCA, right? So your body pH. Mm, okay. So your blood pH is 7.4. If this blood pH changes, these cycles cannot perform their functions properly. Due to which what will happen? your ionization of amino acids will change, and folding and stability of your proteins will change. If there is change, small change in your pH, means 1 unit change in your pH can leads to total destroy of your body. Because ఇప్పుడు మన pH బాడీలో 7.4 కన్నా పెరిగినా తగ్గినా ఏమవుతుంది, your proteins which is acting as an enzymes get inactivated. Or proteins will have different structures, right? Primary structure, secondary structure, tertiary structure. Foldings will be different. Based on your foldings, their functions will be different, right? So once this pH change, this will lead to affecting of your structure, which will lead to damage of your function. Got it? So this is about your pH. Next is your buffer. What do you mean by buffer? I have already said several times. What is mean by buffer? For any biological system, or if you want to extract DNA from liver or something, you will be using phosphate buffer. Any idea why we will make any extractions in the buffer? I'll say small thing. It is a, let's say it is some buffer. Okay.
A solution which is added, which doesn't change the pH values.
Yeah, it is correct. So in normal statement, how you will say? You will say that which can resist the changes in pH when small amount of acid or bases are released. When I take a beaker and if I try to denature or crush my cell, I'll take one liver cell and I will try to crush it. Now what will happen? The acids which is present will be released outright. The acids which are present in your cell will be released. But if you're performing it in the buffer, what will happen? These acids get neutralized. Okay. It is this buffer is trying to do the resist. Keep it in mind or write somewhere. Buffers will try to resist the change in pH when small amount of acids or bases are added. Got it? Now, so if it is a weak acid, what is the components of buffer? It will be made up of weak acid, conjugate base, or weak base, conjugate acid. What do you mean by conjugate acid? Any idea? Conjugate acid will be produced when a base gain a proton. Got it? Okay. First, when you see a mechanism, when there is an addition of acid, see, addition of acid. Acid will have which ions? H plus ions or OH minus ions? H plus ions, right? So this will conjugate base. This will conjugate base. But when there is a addition of base, that will weak acids. Okay, example your acetic acid. Coming to buffer capability and biological buffer. What is mean by buffer capability? The ability of a buffer to resist your pH, right? Maximum capacity, which means pH equals to pKa. Okay, so our normal buffer, when we see bicarbonate buffer, it will be present in, it is the first main blood buffer. Okay, main blood buffer. Got it? So coming to first, remember bicarbonate buffer, which is your main blood buffer. Its pH will be from nearly 7.4. Coming to your phosphate buffer. Phosphate buffer is present in, it will be function used in your cells and for also for urine test. Okay. Coming to protein buffer, where hemoglobin will act as buffer. Okay. Next, it is your buffers. Next, coming to your thermodynamics. What do you know about the first law of thermodynamics, second law of thermodynamics? Any idea about first law and second law of thermodynamics? First law of thermodynamics is energy cannot be created nor destroyed. Okay. Then coming to your second law is entropy will always increases. Entropy of your universe will always try to increase. Next, what is meant by Gibbs free energy? Do you have any idea about the Gibbs free energy? So it can be represented as delta G equals to delta H minus T delta S. Madam, you wrote something, we don't know what the term meaning is. Delta G means free energy change. Okay. Free energy change. Where delta H means enthalpy. Delta T means T temperature. Okay. Where delta S is entropy. Madam, you are saying enthalpy, entropy. Coming to Gibbs free energy, we can understand. Temperature, we know. What is mean by enthalpy and entropy? Any idea? See, when you take any function, okay? It is the heat energy of a system. Enthalpy is heat energy of a system when there is a constant pressure, okay? It will be designated as H, okay? Enthalpy will be designated as H and the units will be kilojoules per mole. If the delta H means if the enthalpy is greater than 0, it is known as endothermal reaction, which means the system is trying to absorb the energy. Endothermic reaction, where if if delta H is less than 0, which means minus. If you get in minus, it is known as exothermic reaction. In the exothermic reaction, there will be a release of energy. Okay. Example, melting of ice will absorb some heat, right? So delta H will be in the positive. So did you get clarity? First, enthalpy means it is the heat energy of a system. So energy can be released or energy can be absorbed by the system, right? So if the delta H value is greater than 0, which means plus 1, plus 2, plus 3 means endothermic reaction. Endothermic reaction means it is trying to absorb the energy. Where if the delta H is less than 0, which means it is in negative condition. So what will happen now? It is an exothermic reaction. So it is require some energy. It will release some energy. Got it? So coming to entropy. enthalpy is designated as H, where entropy is designated as S. Got it? So, it is the measure of disorderness or randomness. Okay? It is the measure of disorder or randomness of the system. Got it? So, how it is measured? It is measured in its units is joule, mole inverse, kilo inverse. Okay, it is the units of your entropy. So, here also same. If delta S is greater than 0, the disorder will increase. Where if delta S is less than 0, the disorderness will decreases. Okay. Example, ice melting into water will increase disorderness. Disorderness ante enti? When you keep a compound, it is an ice, right? Which will be in a filled form when you keep on a plate. But when it is melted, what will happen? It will become water. Disorderness is decreasing, right? Increasing, right? So disorderness means first it is taking very small space, but when there is a melting of your ice, it is trying to occupy most of the space, which is known as increasing in your disorderness. So delta S will be in the positive. Okay. First condition. Got it? Now, what we will see? First, we saw about the pH, then we saw about your enthalpy and entropy, buffer capacity, and thermodynamics. In thermodynamics, you saw about your first law, second law, Gibbs free energy. In that, you saw enthalpy and entropy. Now, we will see about the spontaneous reaction and coupled reaction. What is meant by spontaneous reaction? Students reply. Hello. Hello. So, okay, I'll only say if your delta G, if your delta G is less than 0, it is known as spontaneous reaction, which means exergonic reaction. It is trying to release your energy. If your delta G, Gibbs free energy, is greater than 0, it is a non-spontaneous reaction. Non-spontaneous reaction means unfavorable reaction. Okay. So, if there is an non-spontaneous reaction, what you will do? You will couple it to another reaction which is favorable. Okay. So, coming to couple reactions, it is like you are taking one possible reaction, one impossible reaction, and making your impossible reaction to become possible. Here, cell will couple unfavorable reaction with the favorable reaction. అంటే స్కూల్కి వెళ్ళని పిల్లల్ని స్కూల్కి వెళ్ళే పిల్లలతో పంపడం అనమాట. ఓకే. Example, your ATP hydrolysis. Example, your glucose should be converted into glucose 6-phosphate, right? Which is an unfavorable reaction. So you are coupling with the ATP hydrolysis. When there is an ATP hydrolysis, what will happen? It will release some energy. See here, formation of your glucose 6-phosphate from glucose is an unfavorable reaction. Okay. So you are trying to couple it with your ATP. So what will happen? ATP will break and release one phosphate and ADP. This inorganic phosphate will release some energy, right? It will be utilized to convert into glucose 6-phosphate. Okay. One reaction is favorable and another reaction is unfavorable. Got it? Then your reaction kinetics and enzyme kinetics. What is mean by reaction change, reaction rate? It is change in concentration per unit time, and you will be having different orders like zero order, first order, second order. And what is mean by activation energy? Any idea? Your enzyme, the place at which your enzyme will start producing your product. It is the minimum energy required to start your reaction. Got it? This is known as activation energy. Different reactions will have different types of activations energy. Got it? And coming to your colligative properties, it will depend on number of solute particles. There are types like vapor pressure lowering, boiling point elevation, freezing point depression, and osmotic pressure. Got it? So now we will discuss about your third credit. Okay. I said that your third credit is biomolecules. What is mean by biomolecules? What is mean by biomolecules? Biomolecules means molecules which are present in your body. So they will be divided into macromolecules and micromolecules. What are macromolecules? What are micromolecules? Huh. Okay. Your biomolecules will be divided into major elements, minor elements, and trace elements. Okay. So first coming to your major elements. Major elements which are present in your body are carbon, hydrogen, oxygen, and your nitrogen. Right? Coming to your minor molecules are phosphate, sulfur, calcium, potassium. Right? Coming to your trace elements, iron, zinc, copper, manganese. ఎక్కడో విన్నాం మ్యామ్ గుర్తు లేదు. ఓకే. First, coming to your major elements. I said that major elements are carbon, hydrogen, oxygen, nitrogen. All your biomolecules are mostly composed of your carbon backbone, right? Carbon will be acting as backbone of the organic molecule, and it will form the basic for the carbohydrates, proteins, nucleic acids, lipids, etc. Okay. Coming to your hydrogen, it is your components of water molecule and also organic molecule. Coming to oxygen, it is useful for your cellular respiration. Your nitrogen, which is present in amino acid. If you have a question like how will you identify your amino acid, you will say your amine group, right? Then coming to your minor elements, phosphate, sulfur, calcium, potassium. Then coming to trace, iron, zinc, copper, manganese. First, and what are the organic molecules present in your living system? Any idea? Organic molecules present in your living system. No idea. Carbohydrates, proteins, lipids, and nucleic acids, right? Idea వచ్చిందా? First, carbohydrates will be divided into monosaccharides, disaccharides, and polysaccharides. Monosaccharides means which is made up of only one sugar, where disaccharides means made up of two sugars, polysaccharides made up of more than 2 to 3 units, right? Then coming to your proteins. Proteins are made up of amino acids, right? These amino acids will be made carbohydrates will be having glycosidic bond, where amino acids will be having peptide bond. Okay, so this glycosidic bond or peptide bond are formed by condensation reaction, means due to release of water molecule a bond will form between them. If it is between the carbohydrates, it is known as glycosidic bond. If it is between the proteins, it is known as peptide bond. Okay, and what are the bonds present in the nucleic acid? Do you have any idea what is meant by nucleic acids? Genetic material of our body. What it is? DNA, right? So DNA, RNA is made up of nucleotides, right? Nucleotides are again made up of nitrogen bases, sugars, and phosphate. So you have to see some small news about it. So you have to learn their classification and bonds which are present. And coming to, there are some supramolecular assembly like your ribosomes and chromatin. It will be due to some attraction, a molecules will be formed. Coming to your metabolism, cellular metabolism. What is mean by metabolism? What is mean by metabolism? It is a combination of anabolism and catabolism, right? Anabolism means building process, where catabolism means destruction process. ATP will be destructed. Here, sometimes we will use catabolism for the anabolism. We will break some molecules to build another molecules. Anabolism is building of molecules, where catabolism is breaking of molecules. Sometimes we will break some molecules to build some molecules. Okay. Then uh, now we will see about the structure of your cell wall. There are two types of bacteria based on the cell wall. Do you have any idea? Cell wall is made up of okay, peptidoglycans in the bacteria. Based on the cell wall, they are divided into gram positive bacteria and gram negative bacteria. Gram positive bacteria will have thick cell wall, thick peptidoglycan cell wall, where gram negative bacteria will have thin peptidoglycan, okay, and it will contain a ticonic acid, where negative bacteria does not contain ticonic acid. Here there will be no outer membrane. Here there will be an outer membrane called capsule. Okay. Next, your fourth lesson. We have more, only 21 minutes. Okay. We will jump into carbohydrates. Let's start carbohydrates. What is mean by carbohydrates? It is your fourth unit. What is mean by carbohydrates? What is mean by carbohydrates? Hydrates of carbon is known as carbohydrates. What is the empirical formula of carbohydrates? CH2O taken n times. Okay. It is the empirical formula of the carbohydrates. Got it? So, carbohydrates are divided into, I already said, monosaccharides, disaccharides, and oligosaccharides and polysaccharides. First, if there is only one unit of sugar, it is known as monosaccharide. If there is 2 units of sugar, it is known as disaccharide. If it is 3 to 6 units of sugar, it is known as oligosaccharide. more than 6 units is polysaccharide. Even in monosaccharide, based on your functional group and based on your number of carbons, it is divided again into trioses. Okay, I'll just rub and I'll write here. First is trioses. Tetroses. Pentoses. Hexoses. Heptoses. What is this, madam? We don't know. Trioses means if n equals to 3, it is known as trioses. If n equals to 4, it is known as tetroses. If n equal to 5, it is known as pentoses. N equal to 6, hexoses. N equal to 7, heptoses. Got it? So, based on your number of carbons, they are divided into trioses, tetroses, pentoses, hexoses, heptoses. And based on your functional group, they are again divided into ketoses, aldehyde group and ketonic group. Okay, we will call carbohydrates again as hydrates, polyhydroxy aldehydes or polyhydroxy ketone. What is meant by polyhydroxy aldehyde or polyhydroxy ketone? Now we will see about the trioses. In trioses, based on functional group, I said that they are divided into aldehydes and ketones. Tetroses also divided into aldehydes and ketones. Pentoses, aldehydes and ketones. Same, everything will be divided based on their functional group. First, based on number of carbons, we are divided into trioses, tetroses, hexa, hepta. Based on your functional group, aldoses, ketoses. First, we see about the trioses. Example in aldehydes are glyceraldehyde 3-phosphate, where ketoses is dihydroxy 3-phosphate or dihydroxyacetone. Here glyceraldehyde. Okay. Okay, we will direct this. So in aldehyde, we can write glyceraldehyde, where in ketosis, we can write dihydroxyacetone. Got it? And coming to tetroses, pentoses, hexoses, I'll say about hexoses. Example easy is your glucose and your fructose. Okay. Now I'll just show how can you represent aldehydes and how do you represent ketones. First, coming to aldehyde. Aldehyde functional group is what is meant by aldehyde? CHO. Coming to ketone, C double bond O, right? So you have to remember that. Trioses means number of carbons are 3. 1, 2, 3. In aldehyde, the first group will be aldehyde. Okay, keep it in mind. In glyceraldehyde, it is an aldehyde group. So first carbon will be taking an aldehyde functional group. So second will be OH, OH. Last will be considered as CH2OH. So middle will be H, right? Then coming to your ketone, second position will be taken by your functional group. So first and last will be considered as CH2OH, CH2OH. Got it? It is your glyceraldehyde structure where it is your dihydroxyacetone. So now we will see about the glucose, which is very important. So coming to your glucose, and they are examples of what? Hexoses, 6 carbon, right? So glucose and fructose. What is its formula? CH2O taken n times, right? Here n is 6. So what is its formula? C6H12O6. Now I'll write 6 carbons. 1, 2, 3, 4, 5, 6. Okay. I said that last group will be CH2OH and first will be occupied by your functional group CHO. Next, coming to fructose, again 6 carbons. 1, 2, 3, 4, 5, 6, right? Last will be occupied by CH2OH. First will be occupied by CH2OH, right? So, what is the remaining? You will be writing OH and H. Only at the third position, there is a small change. Rather than that, all will be OH on the right side. If OH on right side, H will be on the left side. If it is H on right, OH will be on left. H. This is the structure of your glucose. Coming to your fructose, I said that second position will be taken by your functional group. Here, 1, 2, 3, 4, 4 bonds are satisfied. And third will be difficult or different. All are same. Got it? So this is your structure of glucose. This is your structure of fructose. And trioses means contain 3 carbon, tetroses contain 4 carbon, pentoses contain 5 carbon, hexoses contain 6, heptoses contain 7. Till 3 to 5, there is no change. Everything will be OH here. Only at the hexoses condition or third position will be changed. Okay. And I'll show you how to form a ring structure. There will be a ring formation between first and fifth carbon. Coming to here, second and this carbon. Due to which you will be getting, now we will write 1, 2, 3. 1, 2, 3, 4, 5, 6, right? So, pyranose ring will be formed. There are two rings, pyranose and furanose ring. For aldoses, pyranose ring. This is known as pyranose ring, where this is called as your furanose ring. Okay. So how will you write glucose? Here, when it form a bond, the hydrogens will be removed here and one O will be bond. I'll show you. Okay. Carbon. Okay. Due to which what will happen? OH, H. If all right side compounds will come down. Okay. Here first coming here. OH, OH, H, OH. This will be like CH2OH and H. See here, there is a first carbon, second carbon, third carbon, fourth carbon, fifth carbon. There is a bond between your first carbon and fifth carbon. You got your O bond here, right? This is known as glucose. Where, coming to your fructose, OH will be present here and CH2OH will be here. And again your H, OH and your CH2OH. This is your fructose. Got it? So there are again chain structure and also boat structure. For the same ring, you will be drawing in this structure, which is known as chain structure. If it is like small bended version, it is known as boat version. Got it? So now I'll show how a glycosidic bond is formed between your glucose molecule. Okay. Now see carefully. I'll be flipping your board. Okay. Here there is OH and here also OH, second position, third is hydrogen, again OH, then your CH2OH and your H. Okay. So This is your glucose molecule, right? So see here. I'll draw the same molecule here also. Okay. Now, a water molecule will be released from both of the molecules. So, how it will be formed? So, release of your water molecule. How it will be released? See here. Due to which you will be forming an bond. Water will be released. Release of water is known as which bond? Condensation, right? This reaction is known as condensation reaction. Now see what is the product here. Your water will be released. So now this bond is known as glycosidic bond. Okay. Now we will give name to it. 1, 2, 3, 4 and 5. Fifth carbon and your sixth carbon. 1, 2, 3, 4, 5. This is your sixth carbon. So bond will be like 1, 4 glycosidic bond. Got it? Glycosidic bond is formed due to condensation reaction, which is release of your water molecule, and your bond will be broken when there is an introduction of water, which is known as hydrolysis. So there are only two reactions which are playing important role. One is condensation reaction, another is hydrolysis reaction. Condensation reaction is helping you to build the bond, where hydrolysis is helping you to break the bond. Got it? This is your disaccharide. Example, if you take maltose. Maltose is a disaccharide which is made up of two glucose units. Okay. So you can write this structure for your maltose. Coming to your first, I explained about the monosaccharide. In monosaccharide, they are trioses based on number of carbons, trioses, tetroses, pentoses, hexoses, heptoses. And in that, based on functional group, there are types: aldoses, ketoses, glyceraldehyde, dihydroxyacetone, and another molecules like erythrose, erythrulose, ribose, deoxyribose, glucose, fructose, heptose, pseudoheptose. Next, coming to your disaccharides, it is made up of two sugar units. Like example, your maltose, sucrose. Maltose is made up of two glucose units. Your sucrose is made up of, I'll just rub it. Okay. Maltose is made up of two glucose units by one glycosidic bond, right? So lactose is made up of glucose and galactose. Got it? Then coming to your sucrose. Sucrose is made up of one glucose molecule and one fructose molecule. These are the examples of your disaccharide. Coming to your oligosaccharide. What is the examples of oligosaccharide? Raffinose. It is a trisaccharide composed of galactose, glucose and fructose. What it is? Glucose, galactose and fructose. Okay. Then another called stachose. It is a tetrasaccharide which is composed of 2 galactose units plus 1 glucose unit plus 1 fructose unit. Got it? This is all about first your monosaccharide, disaccharide, and now it is your polysaccharide. Coming to your polysaccharide, it is made up of more than 6 to 7 units, right? It is a long chain. What are example? Polysaccharide examples are your